So part one of the PowerPoint on chapters 21 and 22 is going to be looking at evolution by natural selection and also on the big idea one from the College Board, which states that the process of evolution drives the diversity and unity of life. That diversity begins with looking at our human genome and actually every organism's genome, because that's where the diversity is going to come from. Because of recent advances, we can now look at DNA sequences, and geneticists can study the genes directly. By identifying the protein coding genes within a DNA sequence, they're able to put together a database that's called gene annotation. And proteomics is a new study, which is the systematic study of the full set of proteins that are encoded by one genome. Because it's proteins, not genes, that carry out most of the activities of the cell. And these proteins are, as you know, coded for by the sequence of base pairs in the genome. So what that means for bacteria and archaea, we have between 1 to 6 million base pairs, of which some of those are going to code for specific proteins. Whereas in eukaryotes, like most plants and animals, there can be genomes that have more than 100 million base pairs. And again, these are going to be examined to see what proteins they code for. And humans have about 3,000 million base pairs in our genome. This table here shows the different genome sizes and the estimated number of genes found in different organisms. So you can pause this and take a look at that by zooming in or enlarging your picture. But what those genes are doing, again, is coding for a variety of proteins. So the systems approach to biology looks at how these proteins interact and what function do they serve within the biological system of an individual. So these are just a sample of the different things that a protein can be responsible for. So those millions of base pairs, again, are going to be given the instructions to make a protein that will fit into one of these categories, anywhere from mitochondrial function to the replication and repair of DNA. Every protein involved in that process is going to be specified and coded for in the genome. This is a pie graph that indicates how we have such a very small portion of our DNA genome which codes for proteins. So sequencing of the human genome has revealed that only 1.5% of that genome codes for proteins, rRNAs, or tRNAs. So the remaining 98.5% is not making proteins. So what is it doing? And what they found is about 25% of it is coding for introns and gene-related regulatory sequences. So as we know, the expression of a gene is going to be controlled by different regulatory proteins. And so those actually are part of this 25% of the human genome. And then, as you know, the exons are the portion that code for the proteins. The introns are the remaining portion that gets processed when transcription has finished and the messenger RNA is ready to be finalized before it's sent to the cytoplasm. So again, that's only about 25% of the human genome that's actually introns. So we're still left with a very large amount of DNA that is non-coding and that is found between the genes and is not an intron. So different portions of this have been defined. We have what's called pseudogenes, which are former genes that have been um, duplicated at some point or in some way have accumulated mutations and are now non-functional. And we also have repetitive DNA, which results in multiple copies of genes in the genome. About 3/4 of repetitive DNA is made up of transposable elements and the sequences that are related to them. And again, those are also known as transposons. Much evidence is indicating that non-coding DNA, which was previously called junk DNA, actually plays an important role in the cell. So section 21.5 in your textbook is discussing the duplication, rearrangement, and mutation of DNA, which is a contributor to the evolution of the genome. So how do we get all that repetitive DNA and these transposons? What role are they playing? And what role they are partly playing is the basis of change at the genomic level. So mutations which are occurring are what are going to provide the variety and the diversity of different phenotypes that natural selection and evolution can actually work upon. It's believed that the earliest forms of life most likely only had the bare minimum number of genes necessary for survival and reproduction, which means that the size of genomes has increased over evolutionary time, with the extra genetic material providing the raw material for gene diversification. This diversification can happen when there are accidents in meiosis, which leads to one or more extra sets of chromosomes. And that condition, when you have an extra set of chromosomes, is called polyploidy. And the genes in one or more of the extra sets can be the things that diverge by accumulating mutations. And they can diverge without affecting the organism because there's still another set of chromosomes that are the original. But the variation that's occurring in the mutated sets can actually be passed on to future generations and persist if the organism carrying them survives and reproduces. So this is one way that genes with new functions can evolve. One of the things that is more recently believed is that there has been an alteration of chromosome structure over time, and that explains the divergence of species. For example, humans have 23 pairs of chromosomes, and chimpanzees have 24. And following the divergence of humans and chimpanzees from a common ancestor, it's believed that 2 of the original chromosomes of the set of 24 in humans was fused in the human line. And then duplications and inversions that result from mistakes during meiosis or meiotic recombination can explain the continued diversity or divergence. What they have discovered is that comparative analysis between chromosomes of humans and 7 other mammalian species paints a hypothetical chromosomal evolutionary history. So looking at that from a graphical perspective, this is what is believed to have occurred between humans and chimpanzees. That was originally believed that chromosome 12 and 13 in chimpanzees came from the original ancestor. And when divergence occurred, these 2 joined together to form the human chromosome 2. And so that is how we got the reduction from 24 to 23 pairs of chromosomes. And then this picture shows the relationship between the human chromosome and mouse chromosomes.